Good morning, dear students. Today in this section, we are going to see the Watson and Crick model of DNA. As the name suggests, this model is proposed by James D. Watson. He is an English physicist and FHC Crick. The picture is shown here. And let's see what are the features of DNA according to their model. So, according to their model, the DNA composed of a sugar phosphate backbone that we can see in this picture. This blue portion is the sugar phosphate backbone, and that lies outside the structure of the DNA. And from that sugar phosphate backbone emerge that purine pyrimidine bases that we can see here. Purine pyrimidine bases. It projects outside from that sugar phosphate backbone. At what angle? 90-degree angle. 90-degree angle against that central axis that pass through that DNA. With respect to that axis, the angle of purine pyrimidine base is 90 degree. And also these two polynucleotide chains, the sugar phosphate backbone plus that base emerge from that backbone. So in that way, two polynucleotide chains are there and they coiled each other. In what way, what direction? Right-handed manner. Right-handedly it coiled each other. And also these two chains are anti-parallel. Anti-parallel means one polynucleotide chain runs 5' to 3' direction and the other one runs in opposite orientation 5' to 3' in this way. So anti-parallel. As we look to the one end of this DNA, we can understand that what is anti-parallelity. Orientation is opposite. And next point is that regarding the bonds, which is the main bond that hold the bases together, is hydrogen bond. So purine pyrimidine, they form a pair, and with the help of which type of force they are forming pairs through hydrogen bonding. And between adenine and thymine, how many hydrogen bond we can see? 2 hydrogen bond. Between guanine and cytosine, how many hydrogen bond we can see? 3 hydrogen bond we can locate. So that means that those portions of the DNA where guanine, cytosine base pairs are more, those regions will be more stable because here 3 hydrogen bonds are present instead of only 2 between the adenine and thymine. Okay, so the hydrogen bond has a role in giving stability also. And regarding the complementarity, that is the another point, another feature. Always adenine pairs with thymine. So by that, and also guanine pairs with cytosine. Adenine always pairs with thymine. Guanine always pairs with cytosine. So in that way, if we know the base sequence in one polynucleotide chain, we can predict that sequence in the other polynucleotide chain. So that is what complementarity. And that complementarity, that idea is put forward by Erwin Chargaff. His rules are then later known as Chargaff's rules. And what are those rules? That rule says that the number of adenine will always be equal to the number of thymine in the DNA. And also number of guanine always be equal to the number of cytosine. And number of purines totally will be equal to the total number of pyrimidines. So altogether, the entire concept we can write in the form of this equation. Total amount of adenine plus total amount of guanine, which is equal to total amount of thymine plus total amount of cytosine. These are purines and these are pyrimidines. So the number of purines is always equal to number of pyrimidines. So that is the Chargaff's rule. Then regarding the dimension, we have seen that there are base pairs between that two backbones of sugar phosphate. And these base pairs, as we look to one base pair, we can see that always purine, purine base pairs with pyrimidine throughout this DNA. Okay, throughout the entire length of the DNA, purine always pairs with the pyrimidine. And this purine is a two-ringed structure. Two-ringed structure. And pyrimidine is a single one-ringed structure. So by that way, the DNA can maintain the same diameter from its beginning till end. Okay, so that's what. Then what's the diameter? What is the diameter of the DNA? It is 20 angstrom. Then what is the height of one turn? One turn means that includes one complete turn. One complete turn. What is the height of that one complete turn? This is one complete turn. Okay. So, the height of one turn is 34 angstrom. And how many base pairs we can see in each turn? In each turn, show you that in each turn, how many base pairs is possible to locate? It is 10 base pair. All these figures we need to memorize. So diameter is 20 angstrom. Diameter means this measurement, and height of one turn. Height of one turn is 34 angstrom, and in each turn base pairs are present. And also the other point is that the DNA possesses a major groove and minor groove. Major groove means this one is a major groove. Say its diameter is more when compared that of the minor groove. Minor groove we can see here. This is a minor groove. Its diameter is less when compared with the major groove. Why? They present a major groove and minor groove because of the tilt. Tilt happened to the base pace with respect to the with respect to the sugar phosphate backbone or the central axis. There is a tilt in this base pace. Because of that tilt, there present major groove and minor groove. That's all about the structure of or Watson and Crick model of DNA. These are its features. For easily memorizing all these features, I have created a video. Its link I will give as card or the end card. So you can go and refer. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in the Benz.